In this two-part video, we'll be reviewing defense against disease for IB biology. In part 1, we'll be covering the following topics: types of pathogens, primary defenses, blood clotting, the basics of innate versus acquired immunity, phagocytes and lymphocytes, antigens and antibodies, and blood antigens. Pathogens are microorganisms that cause diseases. They typically include bacteria, viruses, fungi, and protozoa, as well as parasites. Although methods of infection and transmission vary greatly, the general pattern is for a pathogen to enter the body, multiply, and then move to a new organism, also known as a host. Unfortunately, this process often disrupts normal body functions, leading to illness, and in some cases, death. Each type of pathogen has unique mechanisms to infect and reproduce within its host. Likewise, the body has various mechanisms to prevent infection and eliminate pathogens, which we'll explore next. The first line of defense against disease includes both the skin and mucous membranes, which act together to block the entry of pathogens. The skin forms a strong physical barrier with its outer layer, the epidermis, containing keratin, a structural protein that resists damage and pathogen invasion. Cell shedding, the loss of dead or damaged cells from the surface of the skin and mucous membranes, ensures that microbes attached to the cells are removed and openings to the skin are repaired. Beneath it, the dermis provides strength and flexibility through collagen fibers, while blood vessels and sensory receptors in this layer support repair, temperature regulation, and detection of injury. The skin also produces chemical defenses such as sebum, lysozyme, and acids that create a low pH environment, making it difficult for microbes to survive. Mucous membranes line body cavities such as the respiratory and digestive tracts, secreting mucus that traps pathogens and particles. Specialized goblet cells produce this mucus, while ciliated epithelial cells sweep trapped debris away from the lungs and airways through coordinated movement of their cilia. Together, these components form both physical and chemical barriers, serving as the body's primary defense system before the immune response is activated. When a blood vessel is damaged, such as in the first visual showing an open or cut capillary, the body initiates a rapid protective response to prevent blood loss. The exposed tissue at the wound site triggers the release of clotting factors from both the damaged vessel walls and circulating platelets. These clotting factors begin a chain of biochemical reactions that ultimately lead to the formation of a staple blood clot. In the second visual, the process has progressed to platelet plug formation. Here, platelets adhere to the exposed collagen fibers of the damaged vessel wall. They become activated, change shape, and release chemical signals that attract more platelets to the site. This creates a temporary platelet plug, which helps reduce blood flow from the injury. However, this plug alone is not strong enough to stop bleeding permanently. It requires reinforcement from the coagulation cascade. In the final visual, the clotting cascade strengthens the plug with a network of fibrin strands. The released clotting factors activate a plasma protein called prothrombin, converting it into its active form, thrombin. Thrombin then acts as an enzyme, converting fibrinogen, a soluble plasma protein, into fibrin, which is insoluble. The fibrin strands weave through the platelet plug, forming a dense mesh that traps red blood cells and other components, creating a stable blood clot. What we think of as the immune system is really a collection of mechanisms and responses that work to protect the body from a variety of threats at all times. The innate and adaptive immune systems are two such mechanisms that differ in how they respond to pathogens. The innate immune system is present from birth and provides immediate, non-specific defense against pathogens through physical barriers, phagocytic cells, and inflammatory responses. A fever, for example, is a non-specific response to infection that involves an increase in body temperature that makes reproduction and metabolism more difficult for pathogens. The adaptive immune system, on the other hand, is highly specific and develops throughout an organism's lifetime as it encounters new pathogens. Adaptive responses involve the activation of lymphocytes, which are present in the blood, as well as lymph nodes. These lymphocytes detect, recognize, and remember specific pathogens, resulting in targeted and long-lasting immunity. Phagocytes, including neutrophils and macrophages, are specialized white blood cells that play a critical role in the innate immune response. They engulf and digest pathogens through a process called phagocytosis. Phagocytes recognize pathogens via surface receptors, bind to them, and then internalize them, where enzymes destroy the invaders. Lymphocytes are white blood cells, with the various roles shown here. Central to the adaptive immune system, B cells produce antibodies that neutralize pathogens, while T cells have various roles, including destroying infected cells and helping to activate other immune cells. The adaptive immune response is characterized by its specificity and memory, allowing the body to rapidly mount a defense against recurring infections. Antigens are substances, often glycoproteins, found on the surface of cells that are recognized by the immune system. Each antigen has a unique structure that corresponds to specific antibodies, a type of protein produced by lymphocytes, ensuring that the immune response is precisely targeted. Antibodies bind to antigens on the surface of cells, causing them to clump together and making them easier for phagocytes to ingest. When a phagocyte engulfs a pathogen, it presents the antigens on its surface to nearby lymphocytes. These cells become activated, a process we will return to in the second part of this video, eventually producing large numbers of antibodies. Antigens are not limited to pathogens, as they're found on the surface of normal body cells. The immune system can generally tell the difference, but human cells may be attacked under some circumstances. For example, blood groups are based on the antigens present on red blood cells, so a person with type A blood has cells with A antigens on their surface, and so on. If a person receives a blood transfusion from someone with a different blood type, the immune system may produce antibodies that will bind with red blood cell antigens, leading to a severe and potentially fatal reaction. Not all blood types are incompatible with each other, however. As this table indicates, a person with blood type A has A antigens and produces B antibodies, so they'd be unable to receive type B blood. A person with type AB blood, however, produces no antibodies and would therefore be capable of receiving any blood type without issue. This concludes part 1 of the key biological concepts related to infectious diseases and the immune system. Please continue to part 2 for more information.